Genome sequencing is a rapidly growing field that has revolutionized the way we understand ourselves and the world around us. It is the process of reading and determining the complete DNA sequence of an organism or individual, providing insight into everything from evolutionary history to genetic diseases. On 14th April 2003, scientists completed the finished sequence of the human genome or genetic blueprint of life, which holds the keys to transforming medicine and understanding disease. In today's video, we'll be talking about genome sequencing, its potential, power, and pitfalls. Genome sequencing has become increasingly efficient and affordable in recent years thanks to advances in sequencing technology and data analysis. Today, researchers can sequence the genomes of individual organisms, entire populations, or even entire ecosystems. This has led to breakthroughs in fields ranging from medicine to ecology and has transformed our understanding of the natural world. The process of genome sequencing involves sample collection. The first step in genome sequencing is to collect a sample of DNA from the organism or individual to be sequenced. This can be done using a variety of methods depending on the source of the DNA. For example, in humans, DNA can be collected from blood, saliva, or other tissues. DNA extraction. Once the sample has been collected, the DNA must be extracted from it. This involves breaking open the cells and separating the DNA from other cellular components. DNA fragmentation. The next step is to break the DNA into smaller fragments. This can be done using a variety of methods, including physical or chemical shearing. Sequencing. The fragmented DNA is then sequenced using specialized laboratory techniques such as next-generation sequencing or single-molecule sequencing. The next-generation sequencing or NGS involves sequencing millions of fragments of DNA simultaneously, while single-molecule sequencing reads the DNA sequence of individual molecules in real time. Genome assembly. After the sequencing is complete, the resulting sequences must be assembled into a complete genome. This is done using powerful computer algorithms that compare the sequences to each other and identify overlaps. The overlaps are used to piece together the complete genome. Genome annotation. Once the genome has been assembled, the next step is to annotate it. This involves identifying genes, regulatory regions, and other functional elements within the genome. Genome analysis. The final step is to analyze the genome to gain insights into its biological function and significance. The human genome, for example, contains over 3 billion base pairs of DNA. Genome sequencing allows us to read and interpret this vast amount of genetic information, providing insight into everything from the basic biological processes that makes us humans to the genetic causes of a number of diseases. In modern times, genome sequencing has come out with great potential and is promising to solve a number of problems. Let us now look at some of the key applications of genome sequencing. Personalized medicine. By sequencing the genomes of individual patients, doctors can identify genetic variations that increase the risk of certain diseases or affect the efficacy of certain medications. This allows them to tailor treatments to the individual, improving the outcomes and reducing the risk of side effects. Evolutionary biology. By comparing the genomes of different species, scientists can piece together the evolutionary relationship between them, shedding light on how life has diversified over time. For example, genome sequencing has allowed us to confirm that humans share a common ancestor with chimpanzees and has helped us understand how different species have adapted to their environments. Agriculture and livestock breeding. By sequencing the genomes of crops and livestock, scientists can identify traits that are desirable for breeding programs such as disease resistance, yield, or nutritional value. Forensic science. By sequencing DNA samples from crime scenes or other sources, forensic scientists can identify suspects or victims and help solve crimes. Microbiology and biotechnology. By sequencing the genomes of bacteria and viruses, researchers can identify the genes responsible for antibiotic resistance and virulence. This information can then be used to develop new treatments and strategies for controlling the spread of infectious diseases. The development of vaccines for Covid-19 was also done using the principles of genome sequencing. The power of genome sequencing extends beyond just study of individual organisms. By sequencing the genomes of entire ecosystems, researchers can gain insight into the complex interactions between different species and the environment. This field, known as metagenomics, has the potential to revolutionize our understanding of how ecosystems function and how they can be managed to promote sustainability. While genome sequencing has revolutionized many fields of biology and medicine, there are still several pitfalls and challenges associated with it. Let us now look at these challenges. Technical errors. Genome sequencing is a highly complex process that involves a number of steps, including sample preparation, DNA extraction, library construction, sequencing, and data analysis. Any errors that occur during these steps can result in inaccuracies in the final genome sequence. Coverage and completeness. Genome sequencing technologies have limitations in terms of the amount of DNA that can be sequenced in a single run and the length of the reads that can be generated. This can result in incomplete or low coverage genome sequences, which may not accurately represent the true genome structure. Assembly errors. Genome sequencing often involves the process of genome assembly, which involves piecing together short DNA fragments into larger contexts and scaffolds. This process can be challenging, particularly for genomes that are highly repetitive or have complex structures. As a result, errors can occur in the assembly process, leading to misassemblies and chimeric sequences. Genetic variation. Genome sequencing can reveal genetic variation within a population, including single nucleotide polymorphisms, copy number variations, and structural variants. However, accurately identifying and interpreting these variations can be challenging, and errors can occur in the process. Ethical and social considerations. Genome sequencing raises a number of ethical and social considerations, including issues related to privacy, data sharing, and informed consent. It is important to consider these issues carefully when conducting genome sequencing studies. In conclusion, while genome sequencing has enormous potential for advancing our understanding of biology and medicine, it is important to be aware of the limitations and challenges associated with this technology and keep the ethical criteria into insight. This was all for this video. If you liked it, do not forget to like, share, and subscribe to our channel in depth. Thank you for watching.